Well, hello, and thank you to the organizers inviting me to give an update on our research in MSA here at Mayo. My name is Wolfgang Singer. I'm an associate professor of neurology at Mayo Clinic in Rochester, Minnesota, and I have a longstanding interest in MSA and related disorders. I'm planning to do two things today, namely first cover updates of our work on spinal fluid markers to help with an earlier diagnosis, and then give you an update on our work on stem cells as a disease-modifying treatment. We are now in the midst of a phase 2 efficacy trial, and I plan to go over the rationale for doing this, the study design, and the trial timeline. At this time, the diagnosis of MSA is still mainly based on patients' symptoms and the impression of a skilled neurologist, which receives guidance from consensus criteria that experts in the field have agreed on, and you just heard about their recent revision. There are currently available tests that can help support the diagnosis, which include autonomic function testing, as well as imaging studies such as MRI and DAT scans, but characteristic findings are often not present until the disease has already progressed to a later disease stage. And therefore, it is not really surprising that the diagnosis of MSA usually does not happen until years after symptom onset, when the disease is often already rather advanced. I'm sure you all know about the long list of failed trials that aimed at slowing disease progression in MSA. And I've listed some of the larger ones here, which do include one exception, and that is a Korean trial using stem cells. My team at Mayo has since become quite interested in the use of patients' own stem cells, and we have pursued our own studies using this approach, which I will cover in some more detail in a few minutes. It is important to note that the long list of failed trials in MSA is really intriguing in that there's good scientific rationale for these agents, and concern has rightfully been raised that these trials may have failed because patients are diagnosed too late when the disease has already progressed too far for these therapies to actually take effect. And that aligns the notion that better tests to help in making the diagnosis more securely at an earlier stage are truly needed. And exactly that has been the focus of my team's work in recent years, as we explored various potential diagnostic markers in the spinal fluid of patients with MSA. I covered this topic at this conference last time, so I will focus on the main findings and more recent updates here today. Of the markers we studied, it turned out that there were two particularly intriguing. The first one is called neurofilament light chain, or NFL, which has been shown to be elevated in other neurologic disorders as well, and it is those with very fast disease progression. And that marker allows us to measure the degree of nerve cell damage. In over 120 well-characterized patient samples, we could show that this marker not only provided perfect separation from healthy control subjects, which is shown here on the left, but also separated patients with MSA almost perfectly from related conditions such as Parkinson's disease and certain dementias, and that's shown here on the right. The other remarkable disease marker we found is the detection of misfolded alpha-synuclein protein, which ultimately underlies the pathophysiology of MSA. Now, this was initially pioneered by colleagues in Texas using technology that allows us to detect misfolded protein by mimicking in the vial the progressive accumulation of misfolded protein that happens in the brain. Now, we found in our large cohort that this assay resulted in abnormal protein aggregation only in patients with MSA, Parkinson's, and certain dementias, but not in healthy controls. But even more importantly, the aggregation was different between MSA samples and the other conditions, and that allowed us to separate them completely. Now, new since my last update is that we expanded these studies to patients who only had autonomic symptoms, but no Parkinsonism or balance problems. In other words, they were at risk of developing MSA or similar conditions, but did not fulfill diagnostic criteria yet, and we followed them over time. Now you can see that there's 5 patients out of 32, and those are indicated here with red dots, that had markedly elevated NFL levels at that early stage and also showed abnormal protein aggregation at that stage, suggestive of MSA. And all 5 later developed MSA, and none of the other patients did. Based on that, we concluded that we have 2 markers that alone and in combination have diagnostic value for MSA that may be superior to any other currently available marker, and that our findings really go beyond disease confirmation and allow prediction of a future MSA diagnosis before the disease actually is fully developed. We do believe that these markers have important implications not only for disease confirmation and prognosis, but also for future trials of disease-modifying therapies, which now could be tested at a much earlier stage. Now, the second part of this talk will focus on telling you some more about our currently ongoing phase 2 study of mesenchymal stem cells, and I will cover the rationale, study design, and the timeline. A research group around Phil Lee in Seoul, Korea, reported back in 2009 significant benefit of mesenchymal stem cells in MSA in an open-label study, which later was followed up by a randomized placebo-controlled trial with the hypothesis that mesenchymal stem cells can slow disease progression in MSA through their ability to differentiate into different cell types, including glia-like cells or neuron-supporting cells that can secrete factors that we call neurotrophic factors, and by doing so, have neuroprotective effects. That rationale was previously suggested also based on preclinical studies in a mouse model of MSA. Now, that group randomized patients to receive intra-arterial injections of stem cells into the carotid and the dominant vertebral arteries, and that was followed by IV infusions. And those patients were then compared to patients who underwent sham procedures, or basically placebo procedures. And the study showed indeed slower disease progression in the actively treated patients. Now, this trial, therefore, represents the first and thus far only controlled interventional trial in MSA that actually showed efficacy. And in an editorial, this intervention was called a potentially important step forward with intriguing mechanistic potential, but there were also concerns, a number of concerns actually expressed. Those concerns were that the study was small, that the study was limited to patients with MSA-C. There were also no significant findings when they looked specifically at patients' symptoms and function. And also there were concerns about those cells crossing the blood-brain barrier. But most importantly, there were safety concerns in that over 30% of patients were found to have ischemic lesions on MRI or mini strokes, likely related to the intra-arterial injections. Now, we were intrigued enough by the rationale and the findings that we decided to pursue our own studies of mesenchymal stem cells in MSA, but we went with what we thought would be a safer route of cell delivery, namely injections of cells directly into the spinal fluid through lumbar puncture. At least in theory, this has the additional advantages of solving the problem of crossing the blood-brain barrier, and also provides more widespread access to the central nervous system, which is important because we know that important pathology is present not only in the brain, but also in the spinal cord. Now with that, we pursued a dose escalation safety study in 24 patients who received between 10 and 100 million autologous stem cells per injection, and we followed patients for 12 months. Our primary aim with that study was to assess safety and tolerability, but we also included measures of efficacy and target engagement. We found that the approach is safe and well tolerated at the low and medium dose, and found problems with back pain and some reactive changes at the injection site at the highest dose level, which we therefore decided to abandon. But most exciting were the clinical efficacy signals we saw with a dose-dependent, slower disease progression compared to a matched comparison group taken from our own rifampicin trial. Now, these clinical observations were associated with measurable increases of neurotrophic factors in the spinal fluid, as we had hypothesized. And when we further analyzed the association, it became clear that the lowest progression rates occurred not with the highest, but with moderate or intermediate levels of these neurotrophic factors. And because of that, we decided to add on an additional cohort to our study to explore a cell dose that's between the original low and medium dose. And that would be 25 million cells per injection. And that dose was very well tolerated with essentially no side effects seen. But on the other hand, there was unusual efficacy signal in that there was virtual arrest of disease progression during the year of observation. Also, we found a significant decline of a marker of axonal degeneration that I've mentioned before, namely neurofilament light chain. And that provided additional biomarker evidence of target engagement and efficacy. We felt that these data provided compelling rationale to develop this approach further. And we feel that we have identified an optimal cell dose, and that what we need now is a determination of the optimal dosing frequency, as well as obviously blinded efficacy data. And that has led to the design of a phase 2 trial, which received funding support last year through the FDA Orphan Products Grant Program, and with additional generous support from Mayo's Department of Regenerative Medicine. And this is what the trial looks like. It is a double-blind, sham-controlled trial with placebo being the intrathecal injections of lactated ringer solution, basically a salt solution, and the active treatment being 25 million mesenchymal stem cells that are suspended in that lactated ringer solution. The trial uses an adaptive design, and it starts out with 3 arms where patients will receive either 25 million cells every 3 months, or they receive 25 million cells every 6 months and actually get placebo at 3 and 9 months, or placebo with each 4 of the injections. Therefore, there is a 2-to-1 chance of receiving the active treatment. Now, there will be a recruitment hold after half patients have been injected, and that will be followed by an interim analysis that will look at futility on the one hand, the need to possibly repower or increase sample size on the other hand, and if everything looks promising, we will pick a winner of the 2 active arms, and then resume the study with only 2 arms, namely the winner active treatment and placebo with a 2-to-1 randomization. Now, for very compelling logistical and financial reasons, we had to give up the original idea of a multi-center study for the phase 2, and instead we are conducting this study only at Mayo Clinic in Rochester and plan to enroll a total of 76 patients over 3 years. Now, here's a study timeline that gives a sense of the frequency of study visits. There's a screening visit during which a fat biopsy occurs that is followed by an approximately 2 months of cell production. And then there are 4 treatment visits at baseline, 3 months, 6 months, and 9 months. And during those visits, we will also assess various other study endpoints, including efficacy. And then there will be a 12-month follow-up visit as well. There will also be a 1-week follow-up visit, which is for safety reasons after each treatment, but some of those will be done by phone. The study, therefore, will require a total of 8 in-person visits, and the total duration of the study for an individual patient will be about 14 months. Here is the test schedule, just to give you an overview of procedures at each visit, which include clinical outcome measures, blood and spinal fluid collection, and MRI of head and lumbar spine. Now, the primary endpoint is the rate of disease progression using the change in the Unified MSA rating total score over 12 months. Secondary endpoints include progression in the UMSARS or Unified MSA rating scale subscales, progression in an abbreviated UMSARS version that uses selected items of the original UMSARS that reflect clinically most meaningful aspects of the disease that was per FDA guidance. And then we will also look at progression of autonomic symptoms, at the rate of atrophy of selected brain regions, and other measures on MRI. Now, here I have listed the N and exclusion criteria. And just want to emphasize that we are looking to enroll patients up to 70 years old who fulfill criteria for probable MSA, but are still in an early disease stage with an UMSARS-1 score, not including the erectile dysfunction item, that should be no higher than 17. And patients also still have to be able to walk unaided. Now, I would like to emphasize here that the requirement of an early disease stage is often frustrating to researchers and, of course, also to patients, but there are compelling reasons to do that. Number one is that it has been shown that the outcome measure that is currently available to us, which is the UMSARS scale, has a ceiling effect, which means that progression appears slower as the disease advances. And that's not because it is, but because the way we can measure disease progression is limited. For example, once a patient is confined to a wheelchair, the item gait has already reached its highest rating, and any further progression is not tracked. Also, we have shown in a compassionate extension study to our original stem cell treatment trial, in which we have injected patients every 6 months for several years, that the effect of injections seems to wane as the disease progresses. So compelling reasons why we have to be so strict with inclusion criteria. And again, I understand how frustrating that is. Lastly, I wanted to emphasize that we have gone through a lot of effort to ensure that blinding of this study is optimized to the extent possible. For every patient and every injection, we actually produce both a stem cell product and a placebo product. And randomization will not occur until all release criteria of the cell product are met, which allows us to communicate with the stem cell lab up until the time of injection without risking unblinding. Now, the person delivering the cells to the patient's room is blinded, and the product arrives in concealed syringes. In terms of clinical blinding, we have complete separation of personnel that assesses and manages adverse events on the one hand, and personnel that assesses study outcomes on the other hand. In fact, we have implemented a warning message when a rater of the study by accident accesses a patient's medical record. And all such access is traced and will be audited. And we also have procedures in place to ensure complete blinding of the quantitative MRI imaging analysis. And there will be both a blinded and unblinded statistician from different institutions to ensure that data management and analysis are independent of clinical personnel. And here a quick status update as of earlier this month. We enrolled the first patient into the study in late December of last year. And now, almost 9 months later, we are at a stage where we have enrolled 21 patients, of which 16 have already received at least their first treatment. We had no dropouts to this point, and we have not seen any serious adverse events. On the right here, you can see the enrollment and injection chart. And as you can see, we have been able to really linearly increase the number of patients enrolled, and we hope to continue to do that at a rate of 2 to 3 new patients per month, which gets us to an estimated time point where we reach enrollment for our interim analysis at May of 2023. So about 7-8 months from now. Now, we do want to continue at that rate and definitely welcome referrals, be it physician or self-referrals. We have a very nice study coordinator team that can be reached at adcresearch@mayo.edu. You can also email me directly or call the phone number that's listed here. We get back to everyone who contacts us. We likely will ask you for more information. We may ask for medical records to review, and from there may proceed to a video pre-screen visit, or we may invite you for an in-person clinical evaluation first to secure the diagnosis, depending on the situation. I thank you for your attention, and I'm looking forward to answering questions in the live session.
Hello, everyone. I'm Dana Vosman, instructor of medicine at Brigham and Women Hospital and director of genome platform development at Department of Biomedical Informatics at Harvard Medical School. And it's a pleasure to be here today and to tell you about the applications of genetics in MSA. And I would like to start from telling you a little bit about the personal genome. So each and one of us have 3 billion letters, DNA letters, that we inherited 1 copy of letters from our mom and 1 copy from our dad. And every person is different from another person by about 4 million letters, as you can see here. And this difference is called genomic variant. And since it's quite difficult to understand the differences by looking at 4 million variants or so, we are usually trying to narrow down the count of these differences by looking at smaller number of variants. And in this case, we are looking at 50 to 80,000 rare variants with allele frequency lower than 0.5% in the general population. And every person has between 60 to 100 personal variants that are not seen in any other individual. And to narrow down the count even more, we would like to focus very often only on the protein coding variants, and that would be about 1% of our personal genome. And as you know, many genetic diseases are caused by genomic variants or mutations and environmental exposures. And here we are looking at Mendelian diseases such as cystic fibrosis or sickle cell anemia. And the Mendelian diseases are caused by a very rare mutation with large effect and high penetrance, meaning that if an individual has the mutation, it's very likely that the disease is going to manifest regardless of the environmental exposure. And we are also looking at common genetic diseases such as type 2 diabetes and rheumatoid arthritis. And here we are looking at genome wide association studies where we are looking at common variants of cases and controls and are able to find the association according to statistical significance of the difference between the cases and controls. Um, those are the type of genetic diseases that we are looking at. It's very hard for us to identify rare genetic mutations that have very low penetrance. And also, it's highly unusual for common diseases to have common genetic variants that have high penetrance. So today we will focus on this group, and I would like to tell you about the costs of genome sequencing and the process itself. So the Human Genome Project started in 1990, and the aim of the project was to understand the sequence of the human genome. And it involved thousands of scientists. It took about 13 years of hard work and intensive collaboration, and the cost was about $100 million. And today, in our time, it takes about one scientist to sequence the entire genome. It takes about 1 to 10 days of work, and the cost is below $1,000. So, definitely the time for human genome sequencing is now. But the genome interpretation still remains a bottleneck, with only 25% of rare disease cases being solved. So there is still a need for intensive collaboration in order to identify the genetic cause of many diseases. So this is definitely something that we all are interested to do due to the promise of the precision medicine. And when we look at the generalized medicine, the way that we can simply look at that is that there is a group of patients with similar disease. They have one treatment, one therapeutics, and some of the patients will have therapeutic benefits, others will not have therapeutic benefits and might even have adverse effects. When we look at precision medicine, we are actually classify the patients according to their genetic and other biomarker diagnostics. And then every group of patients will get the right treatment that fits their genomic profile and um, uh, their specific use case in a way that there is a therapeutic benefit for every group in this patient population. And genome sequencing allows tailored therapies for patients with cancer, cystic fibrosis, Parkinson's disease, spinal muscular atrophy, ALS, and other diseases with genetic bases. And today, we would like to ask, can MSA patients benefit from precision medicine? And we hope that the answer is yes. When we look at MSA, the cause of MSA is still unknown. We do know that this is a complex disease, where that means that there are complex genetics involved and environmental exposures. And it's possible that the MSA group has several groups of patients with different MSA subtypes. And we definitely know that one size fits all strategy of the general medicine may not work for therapy. And we are looking to bring MSA to the benefits that the precision medicine promises. And therefore, the MSA genetics has been studied for several decades already. And there are some genetic findings that were established through looking at familial cases where scientists were looking at rare mutations with large effects. And there were genome-wide association studies where scientists were looking for many mutations with small effects, and several genes have been identified in these studies. However, the biological mechanism and the statistical significance of these findings is still not fully established, and there is still more evidence needed for the role of genetics in MSA. And therefore, the MSA coalition established the collaborative cores. And these collaborative cores, as you already heard from others in this conference, have core M for preclinical models, for new models for the disease, core B for biorepositories to collect biospecimen and measure biomarkers, core E to measure environmental exposures and the interaction between gene and the environment, and core G, the genomics core that I would like to focus on today in this talk, that aims to answer the question of, is there a genomic basis for MSA? And the role of core G is truly to understand the genetic basis. And we aim to achieve this goal by assembling genetic information from MSA patients living on 3 continents of North America, Europe, and Asia. We aim to sequence additional genomes from MSA patients with the goal of more than 1,000 genomes to be sequenced. We would like to share a single database that is accessible to researchers worldwide and integrate with ongoing efforts in MSA genomics. And obviously, we would like to sustain the effort by sequencing, by securing government and industry funding. So now I would like to tell you a little bit background about the core G that the MSA coalition established. So there is relationship that is shown on this global map, on this global level. So the MSA coalition core G has collaborations that are established with TGen and University of Florida at the US. We have Sonia Schultz at the NIH that established relationship with European institutions that have access to MSA patients and their samples. There is an established relationship with University College of London at the UK. There is a Japanese collaboration and collaboration with South Korea. And it's important to note that some of these organizations already sequenced the samples by whole exome or whole genome sequencing. So at this point, we can try and establish an access to these databases in order to nucleate them on one platform that can allow access for researchers and clinicians to do research, discovery, and validation of their findings. The National University Hospital of Seoul in South Korea has access to samples that have not been sequenced yet. And our collaborator, Matt Ferrer at University of Florida, has a sequencing platform that enables whole genome sequencing that could be utilized in order to sequence those currently unsequenced samples. And the 3 opportunities that the MSA coalition recognizes is the access to the samples that have been already sequenced and the sequencing of the South Korean samples that have not been sequenced yet. And that's how Core G and the one funding was established about a year ago. I'm very excited to let you know that about a year ago, the MSA Coalition awarded about $1 million over 2 years to those sites that I mentioned in the previous slides. And specifically, Professor Henry Holden at UCL, Professor Bo Jun and Dr. Han Jun Kim at Seoul University of Korea, and Dr. Matt Ferrer and Dr. Matt Hantelman here at the USA. And these data from these sites is going to be sequenced and collected into one single database, which is the clinical genome analysis platform that my team is developing and is already partially developed for analysis, visualization, and access among these researchers, with the goal to be available for access for the global community for research and validation of their findings. We also hope that at this point, while we don't have an access to the samples that have been sequenced in Japan and the NIH yet, we do hope to use these wonderful collaborations in order to validate the findings from the database that we establishing here under the clinical genome analysis platform. So now I would like to show you the cases from South Korea that were sequenced at University of Florida and the cases from TGen as we can find them already analyzed and presented by our clinical genome analysis platform, SIGAP. So you can see here that we have two projects, the University of Florida project with 59 cases so far, many more to go, and the project of TGen with 39 cases so far. Let's look on the TGen cases only, and we can see here the cases, their IDs, and some information that at this point is still focused and facing the genetic expert who is a clinician or a researcher, but hopefully at some point in the near future, we will be able to adjust these interfaces to the patients and their families. So let's look at the first family, and we can see here the patient information, family information, the pedigree, and some status overview. Our workflow starts from accessioning the cases, going to the bioinformatics analysis, filtering the variants from many to only a few, doing interpretation, and finalizing the case. So here at the accessioning step, we can view the pedigree and we can see that the proband is a female with multiple phenotypes accessioned here. And what we can do is display many more phenotypes if we would like as colors. And we can see that none of them segregate with the phenotype of the siblings. We have two brothers and four sisters, and none of them have neurologically relevant phenotypes that were reported by the patient or their neurologist. The parents are also healthy, and this family is very unique because in this family, we were able to collect the samples from all siblings, mother and father, and this information is truly helping with filter out the mutations that are present in the proband but are absent in the siblings. We are going back to the case information, and we can now go to the next step of the bioinformatics analysis. We can review our quality control metrics and the bioinformatics samples. They are the bioinformatics files are available here for download and visualization. And the quality control metrics show that all the quality control metrics met our expectation, having these green flags. And to learn more about these flags and the expected values, we can click at any of these tooltips. After reviewing the QC, we are ready to the most interesting step where we are going down from more than 2,000 variants ingested to the portal to only a few variants that could be then looked at in order to make association between the patient presentation and their genetic profile. So we can see that we can either use our properties for filtering, like maybe gene list, and we are going down to 373 variants, but we can also apply an existing filter set like the recessive and de novo, and this is helping to filter out all the variants that are present in our proband but are absent in our unaffected siblings or the parents according to the potential inheritance mode, which is either recessive or de novo dominant. And we can see that now we are down to seven variants within only a few minutes. And we can now look at these variants, go and learn more about the information on the gene level, the variant level, and the sample level. And we can, after reviewing the information, making sure that the variant is interesting in terms of being technically valid and being biologically interesting, we can go back and move all the variants of interest to the interpretation space. In the interpretation space, we can make interpretation assessments in terms of is it a strong candidate or not. And then in the case review, we can actually check all of our notes and start generating the report if we would like to have a report for research or sometimes even for translational research that could be returned back to the patient under a research IRB protocol. As I mentioned before, we don't know yet whether MSA genetics is caused by one mutation with large effect or several or many mutations with smaller effect and some environmental exposure. We don't know if and what the genetic predisposition to MSA is. So we can also look at the cohorts, not only on sample by sample level. And now, when looking at the cohorts, we can jointly call all the variants of our patients and compare them to healthy controls. So the included samples are summarized here. The statistical analysis is done using statistical tools and statistical tests that are looking at, let's say, gene-based burden test, where we look at genes that have higher number of variants compared to controls. And now we show here the genes according to their statistical relevance within these tests, such as the CMC or VT test. And we can now sort them based on the tests or add additional tests and sort them in this way. We can then go to the cohort browser and we can look at all genes or only at few genes and see how the statistical significance of the corresponding test, it could be CMC or VT, will look like. We can look at the variant density, and we can also look at the visualization of each variance and their significance on cases versus controls. And I would like to finish by providing you with a project overview. So as I mentioned, this is a 2 years award that started September last year and is ongoing until August next year. We are now more or less in the middle. And so far, as you can see, many of our milestones were completed. And specifically, we processed about 30 whole exome families from TGen and analyzed them, as you could see in the demo, in SIGAP. We also processed 5 and then another 55 genomes from South Korea that have been sequenced at University of Florida and analyzed in SIGAP. We are currently building the MSA genome browser for cohort analysis of MSA samples, and we are starting to store the data from last year, and obviously the storage is going to proceed until the end of the project and hopefully beyond. We also hope to sustain this project and build on what we developed already by beginning grant application process for government and industry funds. We are also planning to process another 800 samples from South Korea, the US, and University College of London, and store them in SIGAP. And finally, Henry Holden and his team are working on DNA extraction from more than 360 UK cases at the UCL that are going to be processed, sequenced, and stored at SIGAP and included in our analysis. So by the end of this project, we hope to establish a platform that allows access for research, visualization, and database queries for the community that would be accessible and hopefully will provide with new findings and new promises for the MSA patients and their families. And I would like to finish by thanking our SIGAP team. It takes a village to build a platform that could be used by researchers and clinicians and hopefully by patients and their families one day. I would like to thank our Core G collaborators, and these are wonderful global collaborations that are highly appreciated and providing a lot of fruitful and intellectually stimulating discussions and ideas. And of course, I would like to thank the patients and the families and thank everyone here for your attention today. Thank you.